Hello, everyone, and welcome to the latest in the Royal Society of Medicine COVID-19 series on virus mutations past, present and future. My name's Andrew Jack from the Financial Times, and it's a great pleasure to host our discussion over the next 45 minutes with Professor Sharon Peacock, the Executive Director and Chair of the COVID-19 Genomics UK Consortium. So we'll have a little bit of a discussion, but then very keen to turn over to you for your reflections and questions. So I'd encourage you, please, from now on to use the Q&A function here to post your queries for Sharon. But Sharon, hello, welcome again to the webinar. Great to see you.
Yes, it's great to be back. Exactly 8 weeks since the last time that we met, Andrew, actually, and chatted on this series. So it's great to be back. Thank you for inviting me.
Pleasure, and a fair amount of evolutions, of course, since then.
Yes.
But for people who might not have been with us last time, coming up to speed with the issues, just give us a few words of introduction about the consortium, how it works, and what its role has been during COVID-19.
Yeah. Well, we call it COG UK for short. It's easier than saying COVID-19 Genomics UK Consortium, and what this is a consortium of people, around 400 people, that have set up a sequencing network across the United Kingdom. So it involves 12 academic centres in universities, the 4 public health agencies of the United Kingdom, and the Wellcome Sanger Institute. So on the 1st of April, we set up a network to start sequencing the virus, with the expectation that there'd be a point in time when we'd really need it. And so we take samples from people who've got COVID-19, both who've gone into hospital and who are in the community, but otherwise have relatively mild symptoms. We sequence around 10% of all of those viruses, and then we look at the genetic code. So we can track the evolution of the virus over time. And the key thing that we do is we provide analytical tools to make it much easier to rapidly interpret that data. And then we provide everything to the public health agencies so that it really drives their own investigations, and so that they can use that for intervention decisions and policy decisions.
And we're talking today a lot about variants, but we should perhaps try and understand a little bit, tease out the difference for us with mutations, with variants of interest and concern, the landscape, a little bit of the terminology here when we're talking about the evolution of the virus.
That's a very good starting point, and it can get really confusing. But a mutation is simply a single change in the genetic material of the virus. It's like a typo. So the virus has just under 30,000 different bases or points in its genetic code that codes for everything that it is, its structure. It's a bit like our kind of genetic code decides how tall we are or what our hair color is. And with the virus, it has 30,000 points in the genome. And a mutation is simply a point where there's a typo. So instead of one particular point in the code saying a particular part of the kind of nucleotide, another one goes in by mistake. It doesn't happen very often, but basically as a mistake. And one point in that typo occurs. Now, the only time that really worries us is when that then leads to a change downstream of the amino acid that's being coded for, and therefore the protein that goes into. A lot of those mutations don't matter at all, they're completely neutral. But when they do have a knock-on effect to the amino acid that's selected to go into a protein, then that has the possibility of changing the structure of the virus and the function of the virus. So bottom line is, a mutation is simply a single change in the genetic code of the virus.
And in terms of the variants that we've been hearing a lot about, of course, the Kent, Brazil, South Africa, what have we learnt now about those variants and how they've been spreading?
Yeah. So first of all, I will just describe what a variant actually means, because a variant isn't the same as mutation. So a variant really describes the whole sequence of the virus, which may contain one or more mutations in the 30,000 points in the code. So we use variants, and there are many thousands of different variants, most of which are of no huge biological interest. But there are some that have emerged that are really quite important because they change the way that the virus could behave. So the variants they become variants of concern if they can increase their transmissibility, if they affect the way our immunity deals with them, or if they affect the lethality of the virus and make it more or potentially less lethal. So I was starting with the UK variant that we call B117. That first emerged in Kent. It's still not clear whether it emerged, if it was first detected in Kent or it actually emerged in Kent. But that's the variant that we have in this country. And since we last spoke, we now know that it has spread across the UK and causes nearly all of the cases of COVID-19 in the country, about 98%. There's not much left over for other variants. It's nearly all the 117. The reason it's done that is because it's very transmissible. So last time I spoke, I indicated that this was more transmissible, and the evidence is very strong that it is. And the estimates vary between 30 to 70%, but it's around 50% more transmissible than other variants that were circulating at the time, hence its success in really taking over the country, if you like. In terms of its other characteristics, the good news is that there's really no emerging evidence that there's a problem with the vaccine that we're using. So I would fully expect that the vaccines that we have in this country are effective against our variant. But the other bit of change that I listened to the podcast last night just to check what I'd said previously. And I said at that point, there was no evidence that our variant changed the outcome of disease, but there is growing evidence actually now that that's not the case. Now, the change is quite small. It's obviously important for those people that are affected. But for example, a recent study that was published in the BMJ just a couple of days ago looked at more than 50,000 people who'd been infected with the UK variant and compared their kind of outcome, whether they died or not, with people who were infected with another variant, i.e. slightly before the new variant was very predominant. And what they found was a small increase in the number of people that died. So this study was actually based in the community. So people who had community testing, you had to be 30 years or more in order to get into the study. And the two different groups, the 117 or non-117, were matched quite carefully for age and other factors. And what this study showed was that of 1,000 people who developed COVID-19 in this study, then the increase in a lethal outcome death rose from 2.4 to 4.1 cases per 1,000 cases. And so clearly so important for the people that are affected, but in relative terms, the increase is actually is relatively minor, I would say. I wouldn't want to belittle, a poor outcome for any given individual, but there is a small increase in the likelihood of death from the variant that we have.
And there's no indications of other changes in the sorts of groups by age, for example, ethnicity, underlying conditions that it affects differentially? Well,
because it causes about 98% of all disease now, it's quite difficult to see differentials in that. And therefore, you'd need to use a different measure of prevalence of infection. And so what we need to do then is sort of bring an epidemiological measure to bear. And that's a different question to the one that you're asking me, which is this variant more likely to affect certain people? At the moment, it affects everybody equally because it's our prevalent. So if it's causing 98% of disease, then it's difficult to measure that. So it comes down to, are certain people more affected than others? And that we know from other studies is certainly the case.
And what about comparing and contrasting, as it were, with both the so-called South African and Brazilian variants then? So they haven't really become significant, at least in this country, but they dominate in some of the other parts of the world.
Yes, well, that's right. And they are of concern. And so the variant that we have in this country largely doesn't contain a particular mutation key called E484K. And that particular mutation worries us because it reduces the efficacy, at least in experimental settings, laboratory settings, reduces efficacy of antibody neutralization. So antibodies that are raised to the virus from previous infection or vaccination. In the laboratory, they appear to be less effective against these variants with E484K, and there's increasing clinical evidence for that too. Now, the important factor about the variant first detected in Brazil and South Africa is that they do indeed contain a combination of mutations that appear to be associated with both transmissibility and an effect on the immune response. And that is, it's like a sort of step up in terms of seriousness. And so, yes, you're more transmissible, but by the way, there's something really important to note here, and that is that the vaccines could be less effective and people could get reinfected. And so they are of considerable importance, but they haven't really found a foothold. And so I looked again last night since I last spoke, I looked at the figures for the South African variant and for the Brazilian variant. So we know that the variant first detected in Brazil, which we call P1, we only have 6 cases in the country that we know about. And so that doesn't appear at the moment to have got a foothold, even though we know that actually it's spread to 26 countries in the world. In terms of the variant first detected in South Africa, that's now spread to 48 countries. And we first detected our first case on the 20th of December. But again, the numbers are quite low at the moment. They're running in around 300 in the UK. So we're watching them very carefully and we're very vigilant. And they are important. I wouldn't want to belittle their importance at all. And in some parts of the world, they are very common and causing significant problems for the population there. But at the current moment, they don't appear to be spreading rapidly in the UK.
So perhaps less transmissible and also less lethal in terms of the data we've seen from Brazil and elsewhere?
I think it's too early to say. A recent paper from a group in South Africa who studied P1 in Manaus suggested that that particular variant might be have an increased lethality, but actually it's also in the setting where there's a great deal of disease. The health service is under great pressure, and they said themselves they weren't really clear about that. But certainly that particular variant and the variant first detected in South Africa look more transmissible. So I think it's difficult to say that the reason that they're not being transmitted here is because they're less transmissible than our current variant. That's one possible explanation. Another possible explanation is that actually we've been in quite a kind of marked lockdown, and so all cases are going down of any variant actually. So we've seen the numbers come down a lot in the last few days and weeks, which is really good news. So if we were to say that the UK variant was more transmissible and it kind of keeps out invaders, I don't think that would necessarily, there wouldn't be much evidence for that to be based on. So we have to be vigilant as ever and make an assumption that at the moment it's quite a contained situation, but that we have to keep watching every day and every week to make sure that it stays like that. And also find out the biological reason why that would be the case.
And instantly, what about in other countries, the identification of the so-called Kent variant? Is it spreading very significantly in some other parts of the world from the best surveillance?
Yes, yes, it has. So it's been reported in 94 different countries. And in many countries where it has been detected, it's now becoming increasingly common. Not in every country, I would have to say, but in many countries around the world, it is increasingly common now. And so, of course, countries that do a lot of sequencing will also detect the variant, but certainly it's really coming up in Denmark, in the US, in Germany, and in France. And so because of its transmissibility, once it's introduced, it does have that advantage over other circulating variants. And so it is the case that the 117 appears to be traveling around the world and really expanding where it lands. That obviously won't be true for countries that have very few cases, but in countries that are still seeing transmission and continued cases, then this particular variant is increasing in frequency.
So just before I open it up to questions, just what about some of these other variants? We've heard about California, New York, and so on. Any indications and concerns around them?
Yes. Well, Public Health England publish a list on the Gov.uk website twice a week, which is very helpful. And they have at the moment for what they call variants of concern and for variants under investigation. And so those other variants are kind of under investigation at the moment. The reports so far look as if they're quite similar in their behaviour to the ones that we've already observed. So a combination of transmissibility, potentially partial avoidance of the immune response. And I think one has to be measured in what you say is not a full breakdown of immunity or vaccine efficacy, but not that by a long way. But there does appear to be mutations in those variants that appear to resist the effect of antibodies in the immune response more than other variants. And so it's like there's this kind of phrase, convergent evolution. So all around the world, the virus is mutating in different parts of the family tree for the virus, but actually all reaching the same conclusion that through survival of the fittest, if you like, natural selection, we're seeing variants that are more transmissible, plus or minus, can actually find chinks in the armor of our immune response.
So I see there's a question here about the so-called Liverpool and Bristol viruses. Any thoughts on those?
Yes. So they are at a very low level at the moment. So they're on the list of variants under investigation. But actually, they don't seem to be expanding in the population in the same way that our variant 117 has. And so they're certainly being monitored and tracked. But at the moment, they are kind of under control. So in particular, the variants under investigation, some of those contain this E484K mutation, which I've already said is associated with reduced immunity. But I'm reassured at the moment that they're very few in number, actually, and fewer in number than the presence of the South African variant. And if you're interested, I can give you the numbers, but actually I would encourage you to go to the gov.uk website to look at the numbers, because that updates it twice a week.
So there's a question here about the risk of recombinations between known and potentially unknown coronaviruses, including SARS and MERS. There was some work, Collins Greenstreet saying here, that was done a year ago, almost now, published by NCBI. Any thoughts on that risk of recombination?
Well, it's very likely that SARS-CoV-2 came into existence because of a recombination event. And what recombination means is that parts of the kind of genome of one virus actually recombine with parts of the genome of another virus to create a new virus. That's how new viruses emerge. And so a recombination event is likely to have occurred before SARS-CoV-2 emerged. And it's that recombination that leads to different biological properties, for example, to be able to infect human beings. So that's important, that particular event in the evolution of viruses. And we know that with influenza, a new strain occurs often when there's recombination in exactly that way. We're looking very carefully for recombination in SARS-CoV-2, but at the moment, the evolution really seems to be going one mutation at a time rather than recombination. So we are looking out for recombination, but the current picture is that we either haven't seen it or it's a very rare event. It would be a significant event potentially, but we at the moment there's little evidence actually that this is occurring in the current virus. Of course, there's time yet, and we'll continue to watch, particularly for recombination over time.
So a slightly related question here: is this the first time in medical history we're faced with variants potentially more sinister in nature, presumably, than the original version?
Yeah. Well, that's a really interesting question. And what I would say is that this is a question of whether things get more lethal or less lethal. And the general trajectory for viruses, such as the common cold, the hypothesis is that the common cold coronaviruses were more severe when they were first emerged, but became less lethal. If we look at bacterial pathogens such as tuberculosis or gonorrhea, they are as important as they've ever been in terms of the way that they affect people, their pathogenic potential, their ability to cause disease. In terms of viruses, well, I would say that I'm just working out how long I speak about this because it's quite a big question. I think that overall, our natural, the viruses that we're used to thinking about, for example, influenza, would suggest that these do not get, they could change their lethality. But overall, what I would anticipate is that it's quite difficult to predict what this virus will do. The question is really getting at, will this virus become more lethal or less lethal? And I would say that we have to wait and see what happens over time, very carefully tracking the genome.
So perhaps there's a few themes here around, well, both either vaccination or natural infection and protection. So does previous COVID infection protect from catching the new, well, a variant, in fact, there's a mutation here. Any sense around that?
Yes, yeah. Well, I think that it's if you had infection or you've had a vaccination, then your chances of getting less severe disease is much better than if you haven't ever had SARS-CoV-2 at all. So a really sort of important message here to say, go and get your vaccination if you can, because it's still likely to be effective, even if it's had reduced effectiveness against variants with particular mutations that affect the immune response. And so, there is evidence that particular mutations are associated with reduced immunity. But my expectation would be that if we're all vaccinated against the current variant that's circulating, and we can get numbers right down so we can see less variants emerging, then if you are exposed to a new variant, then I would anticipate that you would have some level of cross-protection from the antibodies that you have to that virus.
And to be clear, you'd said earlier that certainly the evidence seems to suggest that the current vaccines now being rolled out do protect, including against this current Kent variant.
So yeah, absolutely. I would be categorical in saying that the vaccines that we have at the moment are effective against the variant that we have in this country that's causing nearly all of the disease here.
And equally evidence emerging of similar protection from, for example, the Brazil and South African variants, or still too early to say?
Well, we couldn't say about that in our country. Certainly in countries overseas, the reason that that manufacturers are tweaking their vaccines so that they can be effective against the variant first detected in South Africa is that they do appear to be, at least some vaccines appear to be less effective against the variant first detected in South Africa. So it's going to be a case of, and I said last time I spoke, it's really going to be a kind of cat and mouse game of staying ahead of the virus evolution. So right now, vaccine manufacturers are making vaccines that are effective against the South African variant, and they're actually evaluating those in clinical trials at the moment.
And this, it might be quite complex to tease out, but for example, obviously there's been this big debate with staggered dosing. If someone's only had one dose so far in a 2-dose schedule, or there's extra delays, what implications might that have for protection and indeed for driving variants and mutation?
Yeah, well, we took a decision, well, at least the government took a decision based on advice that we would stage our dosing with a 12-week gap. And really that was to protect the very vulnerable. So if you have a certain number of vaccines that you can roll out, it's better to cover a greater number of people than to give a smaller number of people 2 doses. And actually the level of protection that you get from 1 dose is very high. And so, that was a very good decision when you start to look at the emerging evidence. And in fact, we're moving towards a greater distance between the first and the second dose now, because that appears to give a good coverage on the first dose and excellent coverage on the second dose. So I would anticipate that a first dose of vaccine would give you a good level of coverage against the variant that's circulating in this country, and the second dose will actually boost that effect. So we need to go and get our vaccinations. I mean, looking ahead, the likely scenario is that ultimately we will need booster doses, no matter actually what variants are circulating, because our immunity does wane over time to coronaviruses. We know that that's likely to happen. And rather like flu, we're looking at having a booster dose at regular intervals. That might be every year. But it's possible then that our booster is to a different variant versus the one that's, might be circulating last year. Rather like flu, we're going to have to say, what's the variant that's circulating this year? And what are we going to vaccinate against? And so I think that looking at the variants that, as I say, are very uncommon in this country, but looking ahead to the future, it's likely that we will need to look at what's circulating at that point and then predict the most likely vaccine that would be most effective, just like we do for influenza.
And given at the moment, at least, we're moving gradually down the age profile for vaccination, but a question here, is it likely any variants could target children once all adults are vaccinated?
Well, obviously that would be quite concerning, but there is no evidence that variants target children at the moment. And so I think it's most unlikely to say that a variant would be particularly targeting children versus adults. And we know that children have less marked symptoms and signs from their infection. And I would reassure people to say that at the current state of knowledge, there's no evidence that particular variants target particular populations. I can see how that would be a concern for people, as you say, that the vaccine strategy moves through the age groups. But at the moment, the 117 is our prevalent lineage, and we know how that manifests in people, and we shouldn't be concerned that particularly for example, children might be targeted by new variants. But again, we have to be vigilant. We have to keep watching. We have to keep collecting the data.
Actually, just thinking about the impact, particularly for hospitalized patients, there's a question here about the drug regime as well, and whether that in any way might be affected by the emergence of these new variants, how effective we can continue to treat patients.
Yeah. Well, the drugs the treatments that we would be particularly looking at will be the antibodies, the polyclonal and the monoclonal antibodies that people are given may be given in hospitals. So there are particular therapeutics developing around monoclonal antibodies. There was an announcement today about a new monoclonal antibody treatment and being used early on in infection to reduce the probability that somebody gets severe disease and goes into hospital. There are some variants that actually have got reduced, they have reduced susceptibility to some monoclonal antibodies, but that story is just emerging right now. So again, it's a slightly unsatisfactory answer, but we just have to keep collecting the evidence to see what variants are emerging, how they interact with the monoclonal antibodies that we're developing over time. And so the answer is possibly, but not necessarily could it affect monoclonal antibodies. And again, we've just got to gather more evidence.
I'll come back to some of the issues around variants and spread in a minute, but there's a very popular question here about international collaboration, how effective it is, and if a newer variant emerges, for example, in Russia or China, how quickly would you know?
Yeah. This comes back to, there's lots of different ways to answer that question, but the first way I would approach it is that at the moment we don't have enough sequencing capability across the world, and I think that that's clear. And we need to do everything that we can to improve that situation, because variants are likely to be emerging all around the world, and we only actually have eyes on them in certain parts of the world. So it's good for populations to know what variant they have infecting them. It's also good for the world to know what's emerging and how we might want to change our vaccination. And so every effort needs to be made to increase our sequencing capability. And there are many people looking at that now, including the WHO, governments and non-governmental organizations. And so then the question really leads on to then data sharing. So how do you share data? And that certainly sits with governments and the WHO. To date, there has been a great deal of sharing from many countries, for example, saying that from South Africa and from Brazil and from the US. But I would encourage everybody to say that the only way we can tackle this is to be completely open with our data. So our consortium shares everything we generate, and many people do. So this would be a call to say we must share data freely and openly in order to understand what's happening across the world, because only then will we be in a position to really truly understand what variants are out there and how we respond to that.
Party sharing, and then also, are you seeing a scaling up actually of more systematic, comprehensive surveillance efforts in some other parts of the world, which obviously was an area where the UK did have a particular strength historically?
Yeah, well, yes is the answer to that. So many countries are scaling up rapidly now. And actually many countries were already in a very good position. So the Nordic countries had a very strong sequencing capability, but other countries are actually scaling up rapidly now. So the countries that can afford to do this will scale up. The countries that would worry me most would be those that actually would find it more difficult to scale up, the low and middle income countries. And I think that's where we need to focus our efforts to ensure that they are supported to understand what's circulating in their country.
Because how much of the genome is covered by routine testing, and what's the risk future mutations could arrive in parts that aren't routinely checked and therefore spread unchecked?
Yeah, that's a great question. Routine testing by the PCR assay, the molecular assay that's being performed in the Lighthouse labs, don't detect variants at all. So the first line is, do you have disease or don't you have disease? Now, in many places in the world, there's an introduction of a second type of assay, which is based on the same technology, but it actually picks out particular mutations that are associated with given variants. And so I've talked about E484K. So they zone in to say, is there a particular variant in our population? And it's a very quick way of determining whether you do or don't have variants of concern. And so that is being rolled out in many countries now. However, you'll only find what you're looking for using those sorts of assays. And so I would say that it doesn't really avoid the use of sequencing in at least a proportion of the population. So those assays will only be, as I say, detect what they look for, and we need to keep sequencing, which provides completely unbiased ways of looking at the entire genome to see what's happening over time. So I think there needs to be a kind of multi-pronged approach of testing, variant testing with rapid assays, and sequencing, which obviously takes a bit longer than those very fast assays.
Just on the drivers, Sharon, incidentally, of evolution in variants and mutations, there was some reflection, wasn't there? We were talking last time about, for example, whether it was developing in, for example, immunosuppressed patients, those being treated for cancer and so on, where potentially it was remaining longer in the body, for example, or there might have been other treatments that were kind of triggering transmission and mutation. Any reflections and insights and updates on drivers?
I would say that this idea of convergent evolution, where the virus is getting an opportunity to kind of fine tune itself really to become more transmissible or avoid the immune system. The driver for that, the main driver for that, is a combination of very high rates of disease in a population that has partial immunity. And so the virus has mutations that are completely at random, but they will be selected for if they can continue to transmit in a population. And so those characteristics are going to be selected for across the world where we have very high rates of infection and a partially immune population. But we did talk about people who have affected immune systems. And we're seeing growing evidence, it's a gradual growth in evidence, that people who've got immune systems that are not fully working, so for example, they may have diseases that they're treated for cancer or for blood cancer or similar. So they are more likely to get a virus infection that's difficult to shake off. It's like a smouldering infection that could last for many months, actually, in some cases. And the evidence is really based on case reports at the moment where people have taken virus from people like that and sequenced it over time. So when it's difficult to shake off the virus, really what you've got there is a viral training ground. So it's training to persist in the human body over time, regardless of what the body throws at it, or regardless of the treatments that we might give. So there's some emerging evidence that people who are immunocompromised do represent an environment where the virus can actually kind of take an evolutionary leap, if you like, and that's likely to be the scenario that our UK variant emerged in. So what we're obviously thinking about now is whether that could looking out for whether that could happen again. So what that means in policy terms is that we obviously have to protect our vulnerable population. We always needed to do that, but we need to do it. We need to continue to do that, protecting people who have a problem with their immune competency, to try and reduce infection in those populations, both for them primarily, the importance of their health, but also because it does appear that variants can arise under those circumstances where people just can't shake off the virus.
And a viewer is here saying that you mentioned in January that some people possibly carried multiple variants in their upper airways. Any indications what's been found since then with regard to IgA secretions?
Yeah, so it's actually, there's a recent study out actually this week by a colleague in Oxford, and has been looking at mixed infection. Mixed infection does occur, but actually it's quite uncommon. And the other thing that does occur is that that your given variant that's infecting you can actually change in your own body, so that you get a sort of a mixture of the same variant, but it's very slightly different genetically. And those what's called minor variants also do occur, but at very low rates. And so the straightforward answer is that mixed infection is actually quite uncommon.
And then a question here about thinking going to testing. Any implications for the antibody tests of the new variants? Do they have the same specificity and sensitivity?
Yeah, so our antibody tests should work equally well for variants. What would be of more concern is our diagnostic test for whether you've got disease or not. And so the PCR-based assays, they can sometimes be affected by particular mutations that affect the way that the test works. We saw that with our UK variant. And so in the Lighthouse Lab, some tests, they use 3 what they call gene targets, where part of the test detects the presence of that particular part of the genome. And so one of those targets actually stopped working. It's called an S gene dropout. And so the test wasn't false negative at all. It was still a positive. But what that sort of helped us to understand is that mutations can affect the way diagnostic tests work. And so we're on the lookout all the time for those. And if tests don't operate in the way that we think they should, then those particular viruses get sequenced to see what's going on there. So there's constant vigilance in how diagnostic tests work. But you should assume that antibody tests will work as they're intended to.
Mm-hmm. And there's a quite popular question here, though, about testing, notably now in schools with the return secondary schools, and how PCR tests are not allowed to overrule a less sensitive school-based test. Any thoughts on the credibility of that and the implications actually for continued lockdown or return from school? Yeah.
Well, I would follow Public Health England guidance, and so there was some guidance recently published, I think it was yesterday, looking at whether you need to have a PCR assay in addition to as a follow-on from lateral flow devices. And so I'm going to defer to my Public Health England colleagues in the government in deciding how they roll out testing. So that's not one for me.
Okay, I mean, there's lots of questions here around vaccinations and so on, some of which are perhaps a little bit slightly outside your focus. There's a question here though on mutations. Help us understand the implication of what's been reported about how mutations are typically following similar paths. Is that a sign the virus can only successfully mutate in certain ways? And if so, can we address that in the adaptation or evolution, presumably, of the vaccines to respond?
Yeah. This is something we think about a lot. So has the virus shown us everything that it has to offer so far in terms of mutations? This comes back to the idea of convergent evolution. And so all around the world and in different parts of the kind of the family tree of SARS-CoV-2, the virus is coming up with the same combination of mutations that lead to transmissibility and or affecting immune responses. And so the convergent evolution means that the virus has a fitness advantage in order to do that. So and because mutations are completely random, they occur in a large part of the genome, ones that could transmit more, for example, are likely to be the ones that continue onwards. The question is, have we reached a plateau? And so we had a sort of burst of evolution around November onwards, and we're continuing to see that, new variants being described on a regular basis. But when you look at the changes in those viruses, they're all doing fundamentally the same biological things relating to the immune response and transmissibility. So at the moment, we've reached a situation where viruses around the world are behaving in quite similar ways. But then there comes the question, what else might happen? And so people ask, could the virus become completely resistant to vaccine? What other, could it become more lethal? And our answer to that is, I think it's very unlikely that we'd ever get to a situation where the virus is completely resistant to vaccines, because the vaccine manufacturers have been absolutely fantastic and very innovative in keeping ahead of the curve. So I would anticipate that vaccines will stay ahead of where they need to, but we can't predict the future. And so we talk a lot about, can we predict the future? What are the signals of prediction of the future? We know where we are now, but what's going to happen in a month, 2 months, 6 months, a year? And the simple fact is that we're still learning about this virus. It's a very new virus to us. And we shouldn't, it's really important that we don't make any assumptions about what we know based on previous viruses, because I think that would be an error to do so. So at the moment, we know where we are. We need to keep watching to see what happens next, but we can't predict what's going to happen next and the timing of that. In the same way that it would have been quite difficult to predict the events of November, December, and into this part of the year. They're likely to have happened at some point because we're rolling out vaccines and people are becoming immune. But the timing and exactly what happens next is going to be something to be vigilant about. But I'm sorry to say I haven't got a crystal ball today.
And in theory, at least, do you think it's possible to develop a universal vaccine that could tackle all future variants?
Well, the great thing about a theory is anything's possible, isn't it? So in theory, then yes, and no doubt vaccine manufacturers are considering how they could vaccinate universally. But it's quite clear that the spike protein, that's the part of the protein that interacts with our human cells, it latches onto our receptor and then uses that to get into our cells. That's where much of our immune response is targeted to. And so the antibodies that are generated by the vaccine are targeted to spike because that is exactly the interaction we want to stop, stopping the virus interacting with us in the first place and getting into that. So the approach was absolutely the right one. We'll have to wait and see whether manufacturers can have a kind of a global vaccine, but no doubt they're thinking about that at the moment.
Just casting the net wider geographically again than the UK, I see Roger Kobe is asking about the situation in Brazil, which sounds pretty dire at the moment. He asks, is that mainly the result of the variant, or are there other important factors?
That comes back to whether there's an association or cause and effect. So is it only down to the variant? Well, the variant has contributed in all probability to disease there because disease appears to have, because of the increased transmissibility, they've had a spike of infections. And it's possible that the variant avoids part of the immune response. And so people may have developed infection where previously they might not have, for example, if they've had infection before. But to say that that variant is the sole reason that Brazil is in that situation is actually difficult to untangle because there are other factors involved there. And so I would say it's certainly an important contributor, but I wouldn't like to go so far as to say it's the only reason that they're in the situation that they find themselves in.
An interesting related question to that. So does the Kent variant outcompete other new variants like the Brazilian and therefore give our population some protection against these other variants?
It's a great hypothesis, isn't it? And again, we talk about that quite a lot. If you put the three variants up against each other in a race, who was going to win out? I think it's difficult to say at the moment that that one is going to outcompete the other. And the reason for that is that we've been in quite stringent lockdown. So, you know, the possibility for viruses to spread has been quite limited. So all you can say is that it appears to be the case that the other variants are actually not getting a foothold in this country. But if I put them in a head-to-head race, I wouldn't like to say in any given population, if they all started at the same starting point, which one would end up kind of, you know, across the finishing line first. But what I would say is that we do have a very established variant in our country now that's very transmissible. It's difficult to say whether that's been a kind of like a protection for us. I'm sure that people will be working on that question, and it's an important biological question. But at the end of the day, the fact that we have a variant that can be vaccinated against so that we can drive down rates of infection, that's the key way of limiting variants in our country, because variants will arise in our country as well as elsewhere. They won't simply be introduced. We need to focus on getting cases down through vaccination and doing as we're asked to. And then we will start to see that at least that's one way of tackling the emergence of variants in the UK.
And Sharon, as we move towards our conclusion, but maybe coming back to a theme from the start, the theory says increased transmissibility will evolutionarily be more successful. So conversely, shouldn't increased lethality be associated with less successful evolution, i.e. we should move towards less lethality over time?
Yeah, that you think in theory too, and people have actually talked about that at length, and there was a hypothesis or a model developed which is around trade-offs of, if you are more lethal, surely you wouldn't be necessarily more transmissible. And I think there's a lot to be said for that hypothesis. The problem is that we have seen that the virus has variants have emerged that are somewhat more lethal. And so we've certainly seen an example of that, and there are possible examples of other variants that fall into that category. So I think we need to hold fire on the discussion about the fact that the virus will mutate itself out of existence. It might happen, but I can't predict that. And it's proved, if we were to stand by that hypothesis, it's proved us wrong several times so far.
So Sharon, just finally, thanks very much for taking part. Just sort of actually, there's been a couple of questions to clarify around your point actually about the extent to which people should be concerned by this apparently higher rate of lethality with the new variant. Just give us those numbers again, and how seriously you think we should be concerned about this slight increase, at least.
Yes. Well, just to go back to those figures again, and this was a study that was done, you can find it, it's open access in the British Medical Journal. So in this particular study, a study was done in the community that looked at people who were diagnosed with COVID-19 first in the community, and it was in people who were over 30. And so they looked at people affected by the variant versus other variants. And what they found was that they would expect from 1,000 people who got COVID, that 2.5 of those would go on to die. And in this particular study, it showed that just slightly more than 4 people would die. And so there is an increase in lethality. As I say, it's important for those individuals. The step up is of that magnitude. So in terms of being concerned, I think that I would be more concerned about the overall number of cases actually, because there is a small increase in lethality, but what will cause more damage in our population is if we let infection run riot. That will be the biggest burden on us, because then people, a higher number of cases will lead to a higher number of people going into hospital, potentially a higher number of people dying. So out of the two, lethality or transmissibility, I would say it's the transmissibility that is particularly problematic, because it can transmit around our population, a proportion of whom will get sick. And so, reducing our transmissibility and getting on top of our infection is the key to all of this. So I would say, don't be alarmed about this. The evidence has been growing actually over the last 8 weeks. It does look as if it has a small increase in lethality, but we have a bright summer to look forward to with vaccines and a strategy that helps us to reduce cases in our population.
Well, Sharon, once again, very many thanks for sharing your insights. Apologies, we haven't got through all of the questions, but I think we've touched on some really important and useful themes. So thanks for bringing us up to date. I should say also that the RSM new series, The Health Emergency of Climate Change, begins next Tuesday, March 16th at 6 p.m. So do please join the RSM for that. And thanks everyone for engaging. Stay safe.
Thank you very much.